Some regard him as the most important scientist since Darwin.
It's amazing working for Krag Venture. Every day is exciting.
He himself is only a little more modest.
For the first time now, we can actually design life in a computer, make the DNA software, and create new life forms that have never existed before.
He cracked the genetic code, using himself as the guinea pig.
The scientists around the world keep finding things in my genome and telling me what I'm going to die from.
And he followed that up by creating artificial life. He's been compared to God, and even to Hitler.
These are, these were silly comments.
Now he heads teams of scientists on a vital mission.
What we're doing here is going to change the world.
We are here to celebrate the completion of the first survey of the entire human genome.
He said that he learned from me that 3% of his genome was Neanderthal, and he said it explained all his problems.
My guest this week is biologist Krag Venture. Krag Venture has a passion for speed in the laboratory and on the road. His latest toy is a fully electric Tesla motor car.
The entire car is controlled from here. Everything you do is on here. So I charge this from the sun.
Really? And it accelerates.
So we're going 65 already, so it's 0 to 60 in under 4 seconds. So it's,
Really? Yeah.
And look out, there's no noise other than the tires on the road. It's a computer with wheels and a couple of electric motors. So, it's just really phenomenal what they've been able to do. So,
and it really is part of your philosophy of life.
We're trying to put actions into our life. I still love petrol engines, this is my way of balancing my other sins.
This is your dream car, in a way.
It's actually pure driving pleasure.
Yes.
It's not like you're driving some old tank full of batteries or something. It's one of the nicest driving cars I've ever had. It's not short of power, acceleration, or anything.
At the J. Krag Venture Institute near San Diego, teams of scientists are building on the original mapping of the genome. The information in our genes covers every aspect of our bodies, from eye color to susceptibility to disease. Mapping those genes and identifying their individual functions should one day enable doctors and scientists to develop drugs and procedures to target problem genes. From cancer to global warming, to fuel and food supply, the Venture Institute may offer the world new hope. Krag Venture leads from the front. When he mapped the human genome, it was his own genome. You found things about yourself, predictions about yourself, that you didn't know. You learned about what things?
Being the first one to have my genome sequenced, and it's been on the internet now for over 12 years. Scientists around the world keep finding things in my genome and telling me what I'm going to die from. But it's almost entertaining now. It's clear I'm not going to die from childhood diseases anymore. So we don't know how to interpret the data yet in a meaningful fashion. But what it does, it makes it very personal instead of just having an average or a statistic that from a blood test on something that here's the range. And as you know, the ranges are always huge for any value. But when it's just you and your genome, and we can eventually tell you what all that means, then it couldn't be more personal than that. I mean, it doesn't matter where anybody else is on the planet, it matters what yours is.
And you found that when you first found out the facts about yourself?
I've found some intriguing things, some troublesome, but I think it's part of looking at. You know, there's sort of two groups of people I found, those that just really want to know and those that don't want to know anything really fundamentally about themselves. So it's, I think it's just fascinating to look at. What it does, it changes your statistical probability of something happening, which without large numbers, we don't really know what that means. The worst news that I got out of my genome is that I'm heterozygote for the gene linked with Alzheimer's disease. So that means I have a moderately increased chance of getting Alzheimer's disease. But nobody in my family history has Alzheimer's disease. Maybe they died too early. So we need to find the genetic components that help people not get diseases as well. So it's not a prescription, it's not a guarantee, it's not a death sentence or a disease sentence. It gives us information that is hopefully actionable in the future.
Did you change anything as a result of what you learned from the genome?
I've been far more motivated to stay healthy. You know, my father died at age 59 from sudden cardiac death. Those are things I didn't need to read my genome to see, but so I started taking a statin many years ago, lost weight, extremely physically active, you know, trying to do things to change the outcome.
Ever since he was young, Krag Venter has been changing the outcome of his life. At school, a brilliant mind hid behind a rebellious nature.
In school days, there were C's and D's and so on, and you were...
A few F's thrown into it.
A few F's thrown in and so on. But in that sense, who was responsible for that sort of schooling? Was it, and not hitting the targets, as it were, then? Was it your fault or the teacher's fault?
Well, I think I have to take the majority of the blame for that. Later on, I did have some teachers that inspired me, but I was interested. I just don't think I was emotionally and intellectually ready for school. School was in the 50s and 60s. I don't think I'd be emotionally mature enough for it today either. So I just really didn't like it, and I enjoyed doing anything but being in school. I built boats, did all kinds of things. But I'm a late bloomer. And I think I've had tremendous advantages because of that. I argue the school system didn't drive creativity out of me because I ignored the school system.
Unimpressive in the classroom, Krag joined California's beach bar, spending his days surfing the waves and impressing the girls. This was the 1960s. But his hedonistic lifestyle was about to end. The United States was at war. When aged just 20, Krag Venter was called up.
I turned 21 in Vietnam, and actually being in the medical corps and dealing with medicine, both in Vietnam and during my training before going there, it just all of a sudden clicked. Yeah. And I found that the more knowledge you had, the more you could accomplish, the more lives you could save. And all of a sudden, you found out how little was known about science and medicine. And I just really became fascinated with it going forward.
And this thing about life and death is very relevant.
Very relevant in Vietnam, absolutely.
Yeah, that was really dominated your life at that time.
Well, that's for 365 days. That's all I dealt with, sometime between 12 and 24 hours a day, depending on what was going on.
And there was must have been a personal effect when this really hit home to you in a way school had never done.
You know, so it was an important lesson to learn. Fortunately, I learned it as a young man because I think it's shaped my entire life, both mentally, what I've done professionally, not being afraid to take risks, not being afraid to try new things. When you recognize early on that life is very finite, it makes you much more inclined to do something.
You nearly dispensed with life, in a sense, when you swam out to sea and you were going to swim until the point where you were exhausted and you would sink away. Why did you do that, and what stopped you?
Well, you know, being in the midst of having literally hundreds and thousands of guys your age die right in front of you constantly for what was very clear to almost anybody that was in Vietnam more than a day that it was a senseless war. Yeah. And, you know, as a young man, that was a rude introduction to the bigger world. And, you know, you start to lose hope after a while with that kind of situation. But what stopped me is I think I have strong survival instincts.
Yes.
I got out there and said, what the hell are you doing?
And that's when you realized you wanted to live.
Yeah.
But in that second, you weren't sure you could, because you were a long way out to sea at this point.
I had a few challenges to get back, but it worked out all right.
Yeah. But, and that really was the period of the turning point of when you started to really focus on the things that were going to dominate your life from
Well,
then
I
on.
knew at that point I had to go back to school after I got out of the military, and so, you know, I started my basic college education at age 22, not a lot earlier like most people, and I think that gave me an advantage as well.
And after the drowning incident, you quoted, you said at one point, "I wanted my life to mean something. I wanted to make a difference." That was when it was crystallizing what your life was going to be about from then on. And so when you came back from Vietnam, did you have a clear picture of what you were going to do exactly?
Yes, but it's not what I ended up doing. So my my clear picture was to get through college and get into medical school, and so my plan was not really thinking about science in the sense other than learning, but the goal was to go into medicine, and that direction totally changed here in San Diego at UC San Diego, just being introduced to high-end science, high-end scientists, and just getting fascinated by it and actually making a big breakthrough even before I started my PhD training, and I was hooked.
Hooked on science and discovery, but still ready to fight the system, Krag Venter headed for the world of genetics. It was a branch of science which had only really taken off in 1953, when Oxford scientists Francis Crick and James Watson discovered the molecular structure of DNA. The research had taken many years, but Krag Venter was determined that the race to map the human genome should become a sprint rather than a marathon.
Krag is, he's a huge visionary. He has really big challenges, and challenges probably 100 times more than anybody else I've ever heard of or worked with. He has these big challenges that I think even if we can't meet them, but only by 10%, we're still accomplishing something 10 times more than we would have otherwise because of that big challenge. Krag's method of working is incredibly fast. Other labs might take things one step at a time. Krag takes millions of steps at a time.
The actual sequencing took us about 9 months once we were set up to do it. So we started in 1998, and then 2000 is when we announced the first sequence. So it was a good 3 years totally directly involved in it, but it was a very intense time, as you as you recall.
We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind. The moment we are here to witness was brought about through brilliant and painstaking work of scientists all over the world, including many men and women here today.
Today marks a historic point in the 100,000-year record of humanity. We are announcing today, for the first time, our species can read the chemical letters of its genetic code.
What is the result of that? I mean, it can change everybody's future, can't it, really?
Well, hopefully. There's more and more actionable information now coming out almost 13 years later. But it's still early. It's such a complex set of code that we're still in the early stages of understanding even what's in it and what it says. But it gives us the chance for the future to have preventative medicine by understanding what we're susceptible to. So preventing diseases, I think, is going to be the biggest opportunity to come out of this. But there's some nice things right now. A new drug for treating lung cancer that Pfizer came up with actually failed clinical trials until one scientist found there was a single letter change in one gene. And if you have lung cancer and you have that single letter change, the Pfizer drug will work over 60% of the time on reducing that cancer. So understanding these kinds of signals in our genome are going to be a very key part of the future. You know, if you have lung cancer, there's probably no more important information to know whether you have that one letter change or not in your genetic code.
The person, the individual, can find good news about themselves from the genome, and bad news as well. I mean, a terrible prediction about you're certain to get cancer or something like that can destabilize someone.
Yeah. So the good news in this sense is it's all going to be probability statistics.
Yes.
With very few exceptions. There'll be some yes or no answers. But even with the so-called cancer genes that predict whether you get cancer, a lot of people don't get cancer that have those genetic changes.
So people can survive a bad genome report?
Oh, absolutely. In fact, it can give you information to try and do something different.
Yeah.
You know, depends when you find it out, and but also could give you the information to save your life, like the example I gave you with lung cancer.
Yes. But I mean, people say, God, what's going to happen with insurance companies are going to refuse to insure people with a bad genome.
Yeah. So it depends on what country you're in.
Yeah.
In the U.S., there's laws that now prevent that. But at the same time, actuaries that make decisions on risk for insurance companies, it's the exact same statistical information that's going to be used to understand our genomes, and hopefully lower healthcare costs. So in some sense, we want them to use it overall. We just don't want them to use it to exclude people.
Could genome give its mysteries and so on at birth? I mean, can you examine someone's at birth, or do they have to be a certain age before the genome is capable of being perceived?
Well, in fact, one of the things we do here in this building, my team developed single cell sequencing. So we can do a genome on a single cell. So in fact, you can do it well before there's even clearly a life form, just at the early blastocyst stage where you just have a few cells. We can take one out and do the genome at that stage, in fact, to find out if there's a lethal disease. And some people have used this very successfully now when they've had prior children with severe disabling diseases. By doing this pre-implantation selection and looking at the genetic code, you can actually select cells that don't have the damaging genetic changes. We can sequence single sperm cells, we can sequence the egg genome and give couples predictable probabilities on whether they perhaps should avoid having kids in the first place or use these specialized techniques. So it's going to be used, and not just once. If you get cancer, you want the cancer genome to be done.
To demonstrate this, Krag showed me the Robotic Laboratory, where work on single cells goes on day and night.
Turns out every one of our cells is a little bit different. You know, they're not all clones. And so understanding our biology means understanding a little bit about your 200 trillion cells. The assumption was that every one of your cells was exactly the same with the exact same genetic code, and that determined all your different organs. We're now finding when you go from one cell to 200 trillion, there's constant changes. And so what you start with and what you end with are very different things. It's a very different view of human biology and biology than, and you'd never see it if you couldn't do things on single cells. I mean, it's pretty amazing if you just go back 12 years, everybody thought it was going to be impossible to do the human genome, regardless of how many cells you have. Now we can do it in a short period of time on one single cell.
The human DNA, said this report, is 98% identical to chimpanzees. Is that so? There's only 2%.
Well, it's not entirely accurate. When you look at gene regions that code for proteins, sometimes it's even 100% identical. Yeah. Overall, we're about 5% different from chimps when you look at the entire genome. Right. But when you look at specific genes, in many cases, they're identical.
Yeah. Why is that?
Well, it's because we're all part of the same evolutionary tree and pattern. So, in fact, we have identical sequences to most mammals. Mice, rats, cats, dogs largely share the same gene sets with some minor variations because the entire branch of mammals all evolved from an earlier set. So it's pretty stunning how just one letter can change the outcome of somebody's life out of 6 billion. So literally, we're going to find meaningful changes, hundreds of thousands of differences between you and I that affect real outcomes in life. So it seems like when you look at the percentage, it's a small number. When you look at the actuality of all those differences, it's pretty stunning. Your genome is not only your history, but the history of humanity. And so we can find this information and trace it back to the earliest stages. And you know, my genome was used, I think people thought it was cute at the time, to prove, in fact, that our early ancestors actually mated with Neanderthals. Yeah. In fact, I had a lovely honorary degree with President Clinton, and he said that he learned from me that 3% of his genome was Neanderthal, and he said it explained all his problems.
Krag's next venture was to create artificial life.
This breakthrough will have a much bigger impact on humanity than the human genome will.
Some ask, what are the limits?
Will man one day be able to make a new human species?
And now he's also discovering millions of new life forms.
We had no idea how much biology there was on the planet. It's absolutely stunning.
Join us again in part 2.
I'm optimistic we will find life on Mars. We just have to drill down deep enough to find it.
He's a scientific superstar, and now he plans to save the world. Krag Venter's map of the human genome could potentially improve millions of lives. This modern-day Darwin has circumnavigated the globe looking for new forms of life. Krag's team is now attempting to transform the stuff of science fiction into science fact by creating life in a laboratory.
I think in the long run, this breakthrough will have a much bigger impact on humanity than the human genome will. Because it's been the goal of humans from the very earliest stages of developing civilization to get control over nature. For the first time now, we can actually design life in a computer, make the DNA software, and create new life forms that have never existed before.
It has been the stuff of science fiction to bring the spark of life to inanimate chemicals.
It's alive! Oh, it's alive! It's alive! It's alive! It's alive!
Was that supposed to be a clip from my youth?
No, it isn't. In fact, it's not. And it's just touching on the area that you've been involved in and so on, though not as Frankenstein. Did you ever see Frankenstein, the film?
Yeah, oh, absolutely.
Yeah.
And you know, I have actually one of the first editions of Mary Shelley's book, and because it's, you know, it was an important change in literature. So it's and in scientific thinking in the society. So.
In the year 2010, Krag Venter once again hit the headlines with a startling new announcement for the world.
This is the first self-replicating species that we've had on the planet whose parent is a computer.
So the idea of trying to get to a synthetic cell was a means to try and answer what are the basic components of life. Is there a single gene set that we can describe that is fundamental for all life? We found the only way to answer that question was to actually try and synthetically make the chromosome from scratch to put in the components that we wanted to test. But it's taken us a very long time, and in 2003 we came back in a big way, and we started with a small virus or bacteriophage in 2003, and that proved that we could actually write DNA accurately on a small scale. But it took the next 7 years to get it so we could actually get a living cell from making the entire chromosome of a bacteria. So it was a long slog. It took a lot longer than I thought it was, and it would, and it was a lot harder to do.
And this was creating synthetic life.
It was the first synthetic cell that we basically, for the first one, copied most of the genome of a cell that was known. We made some changes in it, but this was the controlled experiment to prove we could do it. So, but it was the first synthetic cell because just changing the DNA in the cell, it changed the species. And I think that's the first time we really learned how absolutely critical the genetic code was, and that you can swap it out like software. The phrase we have is you change the software, you change the species. So we can convert one species into another simply by replacing the genetic software.
At the J. Krag Venter Institute near San Diego, Krag's work on synthetic life is now racing ahead. He describes his team as his orchestra, and that leaves Dan Gibson as his conductor.
Ultimately, I think people will be using this technology to build entire genetic pathways from scratch and restructuring entire chromosomes that they designed at the computer to make better biofuels, vaccines, pharmaceuticals, chemicals, whatever you can think of.
The Venter Laboratory buzz with the excitement of new discovery. And Krag's pride was clear when he showed me around.
This lab is where a lot of the basic research goes on just for synthetic DNA. Come on in. And so they look like labs like any other lab, right?
They
and one of the unique things,
and they look like my desk, too. Somewhat untidy.
You know, about this area of science is you have to have a good imagination. We were talking about earlier, just to do this research, because everything's going on in these tiny little volumes and these little tubes, and you have to constantly carry the whole notion because you can't see what's going on, right? So you have to really think things through carefully. So what's exciting is what goes on in the little volume in the tube. And that changes everything. But that's how our cells work too. You can't see inside the cell and see the DNA, but you know, so it's all the manipulations of changing, cutting and pasting, designing, building new DNA comes from these little what goes on in these tubes where you can't see what's going on.
I think that what we're doing here is going to change the world. It's incredibly exciting. I love coming to work every day.
Exciting to some, maybe, but terrifying to others. Krag's vision of the future has led to some fierce criticism.
If we do not work within nature's system, then nature will fail to be the durable, continuously sustaining force she has always been. We have to put nature back at the heart of the equation.
Prince Charles there has been, for instance, very critical of all this scientific meddling with nature as he sees it, and talks about being swamped in grey goo and so on. Will he in fact have to face up to the fact that there will be living machines with their own agendas during the rest of his life?
Well, he'll certainly have to live up to the fact that we are now not only a society but a globe that is 100% dependent on science for its future. So sticking your head in the sand and trying to hope it goes away and, you know, we can live, you know, out and go fox hunting all the time is not going to be what the future is. We have to have these new tools. We're adding 1 billion people to the population in the next 10 years. And 12 years after that, there's going to be another 1 billion. So that's adding an equivalent of China to the globe over the next 10 years. We're wiping out our environment now to feed and provide energy, food, medicines for the now 7 billion of us. How are we going to add it for another 1 billion and then another 1 billion after that? The existing way we do agriculture, the existing way we do manufacturing, the existing way that we take carbon out of the ground and add it to the atmosphere, they're not sustainable. So environmentalists, in fact, should be the first people to embrace these new technologies because they give us the ultimate hope of changing how we live and keeping our environment.
But criticism didn't come only from royal circles. No less a figure than James Watson himself, the DNA pioneer, also weighed in.
This idea of creating a new form of life, we're just, you know, making a very close mimic to what already exists. So I wouldn't say it's a new form of life at all.
What the camera didn't pick up was James Watson comparing Krag Venter to Hitler for wanting to patent life.
When you heard that quote from James Watson, what did you feel?
Well, he has a long history of making very colorful comments about people. Basically, when we announced that my small team was going to sequence the human genome instead of this $3 billion 15-year government project, which he was heavily involved in, he got very upset because I was taking their prize away. Yes. And said all kinds of irrational things. But I take it in stride because, you know, he's somebody who's made major contributions to science 60 years ago with the co-discovery of the original structure of DNA. And I'm a fan of his for doing that. I think he's made good political contributions to science. But, you know, these are, you know, these were silly comments. And and also he knows we were not patenting the genome, you know. So this was just a means of trying to be critical because we took his prize.
Krag's team are also looking at the medical possibilities of his discovery.
The scale of what you're doing here, you can save the world.
Well, we can provide the tools and hopefully the ability to do that. I think the most exciting application that we have right now is we have a way of making a flu vaccine in a matter of hours instead of months. It took months with H1N1 to get the vaccine. The vaccine came out for the public after the pandemic had already peaked. We now, using our synthetic DNA technologies, in less than 12 hours synthesize the new components for the flu vaccine. But even more exciting, we can actually send biology now to a computer. And this is going to change how we deal with vaccines in the future.
Health benefits are universally good news. But some of the world's senior political figures strongly disagree with Krag about the Earth's buildup of greenhouse gases.
Over the past decade, I've been leading the charge in Washington to make sure the global warming hoax is exposed.
We're not here to serve the Earth. The Earth is not the objective. Man is the objective, and I think a lot of radical environmentalists have it upside down.
But the Intergovernmental Panel on Climate Change, the IPCC, backs up Krag's view that climate change is real and man-made. Average temperatures have risen by almost 1 degree Celsius since 1880, much of it believed to be in recent decades. IPCC scientists now estimate that sea levels could rise between 17 and 59 centimeters by the end of the century. Arctic ice is rapidly disappearing, and the region may have its first completely ice-free summer by 2040 or earlier. An IPCC report in 2007 said that global warming could lead to large-scale food and water shortages and have catastrophic effects on wildlife.
I think, it's clear, at least to most, and at the scientific level, it's very clear that the CO2 levels have been constantly increasing every year.
Yep.
And I think the changes we're starting to see, the water level is coming up. Much more severe storms like the one we just had in the east coast of the U.S. And, as you know, they're talking about creating dikes around New York City, like in Holland, to keep the water out, because the water levels are coming up from all the ice caps melting. So we can't just keep taking carbon out of the ground and burning it and putting it in the atmosphere. So we have to either find ways to sequester that carbon if we're going to keep using oil and coal as our energy sources, or hopefully we can engineer cells to come up with some differences. Carbon is not the problem, it's the source of the carbon. If it's coming out of the ground and we're burning it and putting it in the atmosphere, that's what we can't sustain.
Krag took me to the outdoor labs where he's trying to solve the world's energy needs, and with it, the greenhouse effect. He's growing pools of algae whose genomes have been altered. The idea is to try to make them absorb more carbon dioxide, the main greenhouse gas, and convert it into oil for car fuel.
We have a variety of different projects going on, but most of this is we're trying to find novel ways to grow the algae that we're engineering in the lab. So we're changing the genetic code to try and get algae that get their energy from the sun. Those bubbles were bubbling CO2 in there, so they get their carbon from carbon dioxide. And we're trying to engineer them to produce a whole lot more oil than they would naturally. So we're trying to trick them to get much more obese, so they convert most of their free energy and store it as fat. And fat is just oil, but they wouldn't do it naturally. We thought about just putting them in front of a television and seeing if that worked. It seems to work for humans. But we have to engineer them genetically to do this. And then the goal is, in a very small space, you can produce a lot of oil just from sunlight and CO2. Just be careful not to touch or bump anything, and so here's a very interesting one. This is the one that we're growing to produce a food additive. It's actually an antioxidant. So you can see it's actually a red color. And it's a stress response. So we have to stress the algae so they'll switch all their energy into what we want to switch them to. So it's either to produce fat molecules for energy, here's producing a molecule for making humans healthier. So you can do a lot of different things with it. We can even add light, as you can see over there. Those have artificial light on them. So, it's just a huge range of different things. And if we can, down at the end, it's a whole new system we're trying. I feel it's frustratingly slow, but but that's, we're testing all kinds of novel things here. So it's progressing, but there's been so much hype in this field. A lot of people are promising all these miracle things, and they're going to make fuel in the next year or two from algae. It's just not going to happen, not on an economical, viable scale. So we need probably another 10 years of basic research to really make these big advances in how the cells produce the oil to get it so it can outproduce any plant, outproduce what oil companies currently do.
The leaps forward in science, the excitement of discovery, and the criticism that comes with it. In many ways, Krag is following the footsteps of his hero, Charles Darwin, the 19th century scientist who sailed the globe before arriving at his landmark theory of evolution. A model of Darwin's ship, the Beagle, sits in Krag's office.
It was really a tiny little ship for, but so much of the history of the world comes from, England was a sailing nation, so, but everything was discovered this way. Most of what we know about evolution came from his voyage on this little ship.
Yeah. How long was it, in fact?
A little over 100 feet or so. Oh. But because they were such beamy and deep ships, they could have a large crew on them. Yeah. I'm not sure what his total crew was, but I'm thinking they're on the order of 20 to 40 kind of thing. They were out surveying, and he was just doing biology at the same time.
It's a very good model, isn't it, too? I mean, it looks great. Yeah. So, well, I don't understand, why are you sometimes, to people, controversial about it? Is there a stylistic thing there, or what is it?
I'm not great at suffering fools. And that probably contributes.
Yes.
But I think these are, when you have these kind of ideas, they cause people to think about things in a totally different way. Just showing that DNA is the software of life and that you can make a synthetic cell, that challenges lots of people's concepts of life and what life is, how it works. A lot of these findings from evolution have contradicted people's religious doctrines.
Everything almost that you said there could have been a description of Darwin, rather than a description of you. You know what I mean?
Well, look at the controversy he had, right? I mean, it's
he had that controversy, and there's a real kinship between you.
Well, I think there is with all scientists going. It's not an unusual thing to find scientists who really admire, appreciate everything, because Darwin devoted his life to doing this, from the Beagle to taking the samples to the next 30 years, analyzing it and then writing about it. So other people had talked about evolution and the ideas of it, but he gathered the facts to put together to make the first solid case for it, right? And that's what science is. It's not just ideas, it's converting them into solid knowledge, solid facts.
And now Krag is becoming even more Darwinian. To discover new species, he set sail around the world in his ship, the Sorcerer. His finds were staggering. Around 20 million new genes were scooped up in seawater on his journeys around the world's oceans. Genes which may have potential as sources of new drugs and new life forms.
We had no idea what was even in our oceans. We've gone over 80,000 miles at sea over the last 5 or 6 years on this expedition. Every 200 miles, 80% of the sequences are totally unique to that spot. And we had no idea how much biology there was on the planet. It's absolutely stunning. Most genes now known to science, over 90%, have come from the deck of that sailboat, just taking samples in the ocean and sequencing all the DNA. So it's been a pretty exciting process. We've expanded the net now to sequencing all the DNA that's in the air from microbes that are constantly, we're breathing in and out all the time. And this has basis for the future of exploration of Mars and other planets. It's the ultimate information, and it's blown my mind and everybody else's mind how much diversity there is out there. And we've missed it all. There's more life under the surface of the Earth than there is in the entire visible universe that we see. And that's why I'm optimistic we will find life on Mars. We just have to drill down deep enough to find it.
For all his success and notoriety, one big prize still eludes Krag Venter. Perhaps he'll never win it. It may be the price he pays for his controversial fast-track methods.
When will you get a Nobel Prize? Do you expect one sooner or later, or do you not think about it, or do you think it should have come years ago?
I've had so many scientific awards.
Yeah.
The most important one to me was getting the National Medal of Science from President Obama. You know, for somebody who started out where I did in school, I'm sure I'm the first guy who was a lowly military corpsman to ever get the National Medal of Science in the U.S. You don't do this kind of work for prizes. It's wonderful to have them.
Yeah.
They boost the whole team, but there's no reward better than making these kinds of discoveries. So I don't spend much, if any, time thinking about it, and in part because I've had such nice honors as a scientist, and I can focus on the science.
Krag's determination to find big solutions to global problems means he needs a new laboratory in San Diego. It will, of course, be carbon neutral, driven by sunlight, cooled by water.
We've shown and proven DNA is the software of life, and we can engineer and write new software and get life to do what we want it to. That's been the goal of humanity from the earliest beginnings of civilization. Now we have the chance for the ultimate control, and we really need it badly right now. We have to find ways to provide food, clean water, medicine, fuel in the form of some type of renewable energy on a scale that can't be done today for the population that exists today. But we need these new technologies just to survive without completely destroying the sustenance of all of our life.
I said goodbye to Krag, leaving him in his own futuristic building, a building in which the future of the world could itself be changed.